Food preservation. We know that when cooked food or milk is left in a warm and moist place for a long time, it gets spoilt. Food gets spoilt due to microbial contamination. Most microorganisms cannot grow or multiply when the conditions are dry. Temperature is very low. Temperature is very high. All methods of preserving food ensure that favorable conditions are not provided for microorganisms to grow and multiply. The process by which spoilage of food is prevented using chemical or physical methods is called food preservation. The most common methods used for food preservation are described below. Chemical method. Food can be preserved by using certain chemical substances which can check the growth of microorganisms. Such chemical substances are called food preservatives. Most chemical preservatives remove oxygen from the food item. This helps in preventing microbial growth. Sodium benzoate and potassium metabisulfite are common food preservatives. Sodium benzoate is used for preserving fruit juices and squashes. Potassium metabisulfite is used for preserving cut fruits, jams, jellies, etc. Preservation by common salt. For centuries, people have preserved fish and meat by salting. Meat and fish are covered with dry common salt. Salting draws out water from the food, thus prevents microbial growth and reproduction. Salting is also used to preserve amla, raw mangoes, tamarind, etc. Preservation by sugar. Sugar reduces the moisture content of the food, thus inhibits the growth of food-spoiling microorganisms. Jams, jellies, and squashes are preserved by sugar. Preservation by oil and vinegar. Oil and vinegar prevent the growth of food-spoiling microorganisms. Fruits and vegetables are often preserved by this method. Pasteurization. Pasteurization is a process for preservation of milk. In this method, milk is heated at about 70 degrees Celsius for 15 to 30 seconds to kill the bacteria present in it, and cooling it quickly to 10 degrees Celsius to prevent the remaining bacteria from growing. The milk is then stored in sterilized bottles or pouches in cold places. This method was invented by Louis Pasteur in 1862. By cooling or freezing. Food like fruits, vegetables, meat, and cooked food are kept at low temperature in the refrigerator or deep freezers to prevent their spoilage. Low temperature prevents the spoilage of food because it retards the growth of microorganisms. Canning. Cooked food is first heated to a high temperature to kill bacteria and also its spores to obtain sterilized food. The sterilized food is then sealed in airtight cans to prevent reinfection by microorganisms. By dehydration. Since moisture is one of the main requirements for microbial growth, removal of water from food materials is an efficient method of food preservation. The process of removal of water from a substance is called dehydration. Sun drying is a traditional method of preserving food. Wheat, rice, and pulses are cleaned and dried in the sun. Vegetables like cauliflower, spinach, methi are also preserved in this manner. Advantages of food preservation. It decreases the food wastage by avoiding spoilage. It increases the storage period of food materials. Nutritional value of the food is retained for a longer period. It ensures that food availability in distant places and during off-season. Frozen food items such as suffer vegetables and off-season fruits and vegetables are available in the market throughout the year all over the country. Nitrogen cycle. Our atmosphere has 78% nitrogen gas. Nitrogen is one of the essential constituents of all living organisms. The atmospheric nitrogen cannot be taken directly by plants and animals. It has to be first converted into nitrates, which is the principal source of nitrogen for green plants. The conversion of nitrogen into nitrates is known as nitrogen fixation. Nitrate assimilation. Plants absorb nitrates from the soil through their root system and convert them into plant proteins. The herbivorous animals eat green plants and convert the plant proteins into animal proteins. The herbivorous animals are eaten by carnivorous animals and synthesize animal proteins. Ammonification. When animals excrete waste materials like urea or uric acid, or when plants and animals die, the proteins present in them are converted into ammonium compounds by the putrefying bacteria and fungi found in the soil. This process is called ammonification. Nitrification. Ammonium salt in the soil is converted first into nitrites by Nitrosomonas bacteria. The nitrites are then converted into nitrates by Nitrobacter bacteria. The nitrates so formed in the soil can once again be absorbed by the plants. This process is called nitrification. Denitrification. A part of the nitrates in the soil are converted by Pseudomonas bacteria into nitrogen gas, which escapes into the atmosphere. The conversion of nitrates into free nitrogen gas by denitrifying bacteria, Pseudomonas bacteria, is called denitrification. The cycle is complete. As a result, the percentage of nitrogen in the atmosphere remains more or less constant.